Hello, my name is Zach Howard. For my Biotech Expo project, I was testing the efficiency of plasmid vector versus phage vector in transformation of bacteria. For my project, I did lab research. I was testing the transformation efficiency of E. coli using two different vectors. These two vectors were the bacteriophage and the plasmid. The bacteriophage, depicted here, is a virus that infects bacteria. The virus transduces the DNA located inside the virus into the bacteria, where the bacteria then proceeds to replicate the DNA and produce whatever the DNA codes for. The plasmid is another piece of DNA. It is covalently closed a circle, and the plasmid DNA is transduced or put through the bacterial membrane by a process known as heat shock. The hypothesis of my experiment was that the bacteriophage would transform the bacteria more efficiently. I believe this because the bacteriophage not only coded for the protein I was looking for, which was the beta-galactosidase enzyme, but they also coded for more bacteriophage. These more bacteriophage produced infected more cells located around the E. coli cell that was initially transformed. This led me to believe that the bacteriophage would infect more cells in a faster period of time, being more efficient. The materials used in my experiment and the method used in my experiment was the LacZ assay for blue-white screening. This assay was meant to allow for comparable results between the two different methods of transforming bacteria. By transforming the bacteria with the LacZ gene, they would produce blue colonies, which would indicate that the bacteria was transformed. For the bacteriophage, they would produce blue plaques. The transformation was carried out with both a plasmid and a phagemid. Phagemid being the plasmid DNA that codes for the bacteriophage virus. For the phage infection and transduction, I used a top agar overlay. This means that I transform the bacteria and then mix them with more lawn bacteria and then mix them with a soft agar overlay, which I then poured over a plate. The bacteria I used in this experiment were E. coli K12. Here are two pictures of the DNA maps for both of my plasmids. The plasmid on the left is the PUC19 plasmid. This plasmid was 2,686 base pairs and coded for both the LacZ gene and ampicillin resistance. However, I did not use ampicillin resistance because I wanted to make sure that I knew exactly how many bacteria were both transformed and then not transformed to effectively calculate the efficiency of both. On this side, this map indicates the M13MP18 DNA. This plasmid coded for not only the proteins necessary for viral production, but also the LacZ gene as well for my LacZ assay. This was comparable in size at 7,249 base pairs, which was my attempt at controlling the variable that is how big a plasmid might affect the efficiency of the transformation. So now for my results. The results of the experiment were somewhat inconclusive. Here you can see that the positive M13 plate, which is the bacteria transformed with M13 DNA, this is the control plate. There was no growth. All of the plates were incubated for a full 24 hours and transformed at the same time to allow for control of the experiment. Here, this slide depicts the positive PUC19 plate, which is bacteria transformed with PUC19. This plate was diluted down to 1/100th of the initial dilution, or the initial transformants. As you can see, there are definite colonies, but no blue colonies, indicating a problem with either the IPTG or XCL solution, and therefore no ability to quantitatively analyze the transformation. Here is another positive M13 plate, but this one was not the control plate, so this is plated on both the IPTG and XCL solution, allowing for blue-white screening. There were, however, no blue plaques, although there are plaques located throughout the plate, but they are somewhat dilute. This, however, does mean that there were some bacteria that were transformed because they would need to be transformed for them to produce plaques. Here is the PUC19 initial dilution, which is a full lung. This is completely expected because there was no dilution done with this bacteria, so it should be growing in a full lung. However, there were no blue or white or blue areas, which indicates that that there was a problem with either the IPTG or XCAP solution. Here is my positive PUC19 plate on a tetracycline resistance plate. This is a very important plate because for the bacteria to be infected with the phage, they needed to have a F prime fertility factor. This fertility factor was selected for by plating my E. coli on tetracycline plates. The tetracycline resistance and the fertility factor were located in the same plasmid, meaning that any bacteria grown on the tetracycline plates was viable for phage infection. This plate shows that my bacteria used in the PUC19 transformation were equally similar to my bacteria used in the M13 transformation. This plate indicates the negative M13 control, which were not transformed with M13 DNA. However, there appears to be some plaques and plaque growth on the plate. As you can see, there is definite bacterial growth by these lines here. This is odd, considering that this plate was not transformed and therefore should not be producing phages. This plate is the negative M13 control, or negative M13 plate, which was not transformed with M13, but also plated on the AccuTG XGAL LB plates. This plate has a lot of growth on it, which is expected but no plaques, which is also expected because the bacteria plated should grow, but there should be no plaques present because none of them were transformed with M13 DNA. Therefore, none of them should produce M13 phage. This plate is the negative PUC19 plate, which was not transformed with PUC19 DNA, and it sports some growth in distinguishable colonies, but no blue colonies, which is expected as the bacteria did not contain any LacZ gene, which means they could not produce any beta-galactosidase. So these results are interesting. They're definitely inconclusive because there was no way to successfully quantitatively analyze the results. However, the data does somewhat show and refute my hypothesis because the plate that was, or the M13 plate that was transformed with M13 DNA did have some plaques, which definitely indicates that the bacteria was at least transformed. This, however, can't be conclusive because I could not compare with my PUC19 plate, which had colonies as well, but no blue colonies. So in the end, there was no tangible quantitative data from my experiment, but it did help prove, or it did somewhat refute my hypothesis.